Okay. All right. So tonight we're going to talk about carbohydrate metabolism. So last week it was a general principles of energy production in the body. Now we're going to talk specifically about what happens with carbohydrates that we take in. So this is a high level overview. I decided to kind of start with this just to kind of give you a big picture of where we're going, and then we'll dive into this, and then we'll end up back um, where we'll end up back on this diagram at the end. So down here we see this citric acid cycle, or the Krebs cycle, that we dealt with last week. The principal feeder of this is that acetyl CoA, and it feeds in here, and we end up producing these 26 ATPs at the end of this. This produces that fat H and NAD H2, or fat H2 and NAD H, that then feeds that electron transport chain, and we end up building 26 different ATPs from one glucose. And here's that byproduct of the CO2 that I was just asking you about. But where do we get the acetyl CoA at? Well, one of the sources is from carbohydrates. So we eat carbohydrates. This gets digested into our body, and it mostly gets broken down into glucose. Glucose will then go through glycolysis, which is breaking that glucose down into smaller species, and you will end up with pyruvate. And pyruvate will convert to acetyl CoA and enter into this cycle. So us eating carbs gives us sugars, which then get broken down into ultimately acetyl CoA to go through the citric acid cycle. If we have so we're going to look at glycolysis tonight. But if you have too much glucose in your body and you don't need it, then what your body can do is form glycogen. If you remember, glycogen are these multi-branched chains of all these glucoses. This is where we get fat in our bodies from. So the excess glucose can get converted into these glycogen polymers through a process called glycogenesis. So glyco, like glycogen, genesis, making it. And so this is where it stores the excess glucose as glycogen. As our bodies need glucose, though, so if we're not eating the carbs to get the glucose, or we're like exercising, we're doing work, and we're consuming this glucose, that's where your body is going to peel off glucose from the glycogen to get the energy. So your body's going to go, oh, I need energy. And so it's going to go through a process called glycogenolysis, which will peel off glucose from these glycogen polymers that are stored in your body and produce that glucose, which can then go through and feed this cycle and produce all this energy down there. Does that make sense? Any questions with that? If you're not eating glucose, you're not eating carbs to produce the glucose, and your body needs energy, what can happen is it can take pyruvate and make glucoses itself. And this process is called gluconeogenesis. And the reason why it's called this is it's making new glucoses from pyruvate that's already in your body. So it's not breaking down carbs and getting glucose. It's taking this pyruvate, which is a result of glycolysis, and it's making glucose because it's like, oh my gosh, I need more sugars and for my energy, so it'll cool that off and put it in as glucose. You can see here that this is an expensive process. It consumes 6 ATPs in order to do this. And then we have a excess glucose can also be sent over through what's called a pentose phosphate pathway to make pentose sugars and carbon dioxide. And these can be used to make nucleotides. Or if it does this and then we suddenly need glucose again, it can take this and form triose and fructose and feed that back in to this glucose as well. So this does act as a little bit of a reserve as well in your body if you need it. So we're going to look at all these steps, but it just kind of gives you a high level view. I'm not reading your question, Anna. What would happen first, the body making neoglucose from pyruvate or the body going into ketosis? I'm not 100% sure, but I think maybe this gluconeogenesis is part of that ketosis. That could be that, well, I guess I would say probably, I'm no expert on this, but I'm guessing that probably once you've depleted this pathway, then you're going to go into ketosis because you're totally without any carbs at all. And your body is going to eat up as much of this as possible to keep you alive, right? So that's what ketosis does for you, is it gets you into that state where your glycogen reserves are getting eaten up and you lose weight. So I'm not sure if I answered your question or not, but thank you. That would be my guess. Okay. So the first step here is this digestion step. Digestion is the biochemical process by which food molecules are broken down through hydrolysis into simpler chemical units that can be used by cells for metabolism. Digestion begins in the mouth. We have an enzyme called salivary alpha amylase that causes the hydrolysis of those glycosidic linkages in starch, which is a carbohydrate. And it also will catalyze the hydrolysis of glycogen in meats into smaller polysaccharides and maltose. So that enzyme's already in your mouth and is already at work to start breaking things down. Once you swallow it, it goes into your stomach, but not much carb digestion actually happens in the stomach because there are not the enzymes for it. These enzymes that break this stuff down does not do well with your stomach acid, and therefore they're not able to survive in that environment, so you don't get much carb digestion happening there. The primary site is in the small intestine. So these smaller polysaccharides and maltose get shunted into your small intestine, and that's where it primarily happens. And here's the small intestine. So your pancreas secretes alpha amylase, and this will break down the polysaccharide chains into shorter segments, and then those will get broken down into maltose and glucose. So maltose is 2 glucose units. So you'll get a maltase breaks maltose down to those 2 glucose units. Sucrase will take sucrose, which is made up of a glucose and a fructose, and it will break that sucrose into those 2 species. And then there is a lactase, which will break lactose down to glucose and galactose, which are two components of lactose. These then get absorbed into the bloodstream through the intestinal wall, and it's on these little ridge things on your intestinal wall that are called villi. And this is so they pass through into your bloodstream through that intestinal wall, through those villi. And this does require, remember we said transport of nutrients through the body requires energy. So it does require energy from your ATPs to move those nutrients that it's broken down into your bloodstream. These things get delivered to the liver where glycolysis happens. So here's again kind of an overview of it. So we eat our carbohydrates. In our mouth, we have this salivary alpha amylase, which does some hydrolysis. It begins the process of digestion. You're going to get out of that polysaccharides, maltose, sucrose, and lactose. These will go into the stomach, of course, from your mouth to your stomach. Not much happens in there with the carbs. Other things happen in your stomach, but not carb digestion. We'll see next week things that happen in the stomach. But you do have that stuff moving into your small intestine, where these enzymes from your pancreas will break those down into maltose, sucrose, and lactose. And these will get broken down into glucose, fructose, and galactose. And then those will be transported through your intestinal lining as monosaccharides into your bloodstream. So you're basically taking these large chain carbs and producing monosaccharides. That's why I was saying we're powered by sugar, basically. All the carbs we eat just get powered into sugar or transformed into the component sugars. Any questions there? So glycolysis is the first one we'll look at. So glycolysis is the breaking down of glucose, and ultimately we end up with that pyruvate, which will then get converted into that acetyl CoA and go into the Krebs cycle. So the overall reaction of glycolysis is glucose will react with two NAD pluses, two ADPs, two inorganic phosphates, and it will produce two pyruvates. You will get 2 moles of a reduced NAD plus. You'll get 2 ATPs out of that. So we're adding a phosphate on to that ATP. And we will have 2 hydronium ions left over, as well as water. And the oxidizing agent of all this is the NAD+. It is oxidizing the glucose and itself being reduced. So here is a glucose, and this is pyruvate. So you can see here it's breaking that ring structure out, and we're getting this carbon forming a double bond with an oxygen here with the loss of the hydrogen. This all happens in a 10-step process that we're going to walk through. The first 3 steps are dealing with a 6-carbon stage. And then the last 7 steps happen in what's called the 3-carbon stage. So it's going to go from the 6 carbon in the first 3, but by the time you get to the 4th stage, you're now dealing with a 3-carbon chain. So step one is the glucose gets phosphorylated into glucose 6-phosphate. And this is going to require energy from the ATP. So here's our glucose. We're going to lose that hydrogen, and we're going to take a phosphate ion off of this ATP, and it's going to get tacked on to where that hydrogen was at. And so it's consuming an ATP to an ADP in that process. And there is an enzyme in this as well called a hexokinase that helps this all happen. And just remember, remember that this P with a circle on it is just shorthand for that whole phosphate unit that comes off of that ATP. Step two is we have glucose 6-phosphate getting isomerized into fructose 6-phosphate. And so this is an example of an isomerase. So what happens here is we've got this glucose 6-phosphate from the previous step. And this is going to, with the action of this enzyme, we're actually going to pull a carbon and a alcohol group out of the chain, and we're going to go from 6 carbons in the ring, well, 5 carbons, well, 6 counting that one, I guess, and we're going to go into a 5 carbon ring, and this is now fructose 6-phosphate. So this is fructose, and then it's got this phosphate on that 6 carbon there. So it's going from a hexose to a pentose through this process. The fructose 6-phosphate from the previous step now gets phosphorylated into fructose 1,6 bisphosphate. And so there's now going to be 2 phosphates on here. Instead of just the 1, we're going to replace this hydrogen with a phosphate. This is going to, again, consume energy because we need that phosphate, so it peels it off from the ATP and forms ADP. And the reason why this is called bisphosphate is because instead of diphosphate is because these phosphates are on different places on the ring. So they use the term bis, which is like like bicycle, 2 wheels. Bis is a prefix that means 2. They use it instead of di to denote the fact that these are in different places in the ring structure. So we've now got a diphosphate or a bisphosphate going on here. Oh, I put that in the note. Okay. And that's going to require energy to do that. And again, you're going to have an enzyme that is causing this reaction to occur. This is called a phosphofructokinase. Step 4 is this fructose 1,6 bisphosphate splits into 2 C3 molecules. So here's our end of the first, the 6 carbon set, and we're now going into 3. So here is the fructose 1,6 bisphosphate shown in an open chain form, and it's going to cleave right in the middle here, and you're going to have a dihydroxyacetone phosphate, and you're going to have a glycerolaldehyde 3-phosphate. So this is a dihydroxyacetone, and with a phosphate group on it from this one, and you're going to have a phosphate group on this side from this lower portion of that. And again, there's an enzyme that does this work, and it's called an aldolase. Dihydroxyacetone phosphate from the previous step gets isomerized into another glyceraldehyde 3-phosphate. So we had, remember, one of the products was this. This gets switched to that. So you're now going to have 2 of them. So that's step 5. Step 5 is going to take that dihydroxyacetone phosphate, and it's going to convert it over into this glyceraldehyde 3-phosphate. So we now have 2 of these guys instead of just 1. Step 6 is now these 2 glyceride 3-phosphates get phosphorylated into 1,3-bisphosphoglycerate. And so here we have our 3-phosphate. It reacts with NAD plus and an inorganic phosphate, and we add that into the molecule up here on this carbon at the top. And again, it's bis because these 2 phosphates are in different places in the chain, and you're going to get NADH as a product of that. Step 7 is we take this 1,3-bisphosphoglycerate and we're going to convert it to 3-phosphoglycerate. Now in this case, we're peeling a phosphorus off, so we're breaking that strain bond. Remember that tilde means a strained bond with that phosphate ion. And again, keep in mind this is a phosphate ion with that circle around it is a sort of a shorthand for that. So this phosphate breaks off and will react with an ADP to give us ATP again. So this is an energy producing reaction, and we're going to end up with 3-phosphoglycerate. We're going to have phosphorus still down here, and we're going to have an oxygen with a negative charge now up here, and that's why it's called a glycerate, as it's this ion. ADP converted ATP. Yeah, I guess I covered that. Step 8 is we have this 3-phosphoglycerate from our previous step that is going to have a mutase as an enzyme that will actually switch the phosphate ion here up onto this hydroxyl group here. So it's actually going to switch those around. It's going to flip the hydrogen down here, and it's going to move the phosphate ion up here. Everybody see that? I put a little double arrowed line in there to indicate that as well. So it's mutating this from one form to another. Step 9, we're almost done. 2-phosphoglycerate from our previous step is now going to get converted to a phosphoenol pyruvate by popping off water, and it's going to form a double bond here between these 2 carbons because of the loss of the water. And this is called an enolase because it's catalyzing a reaction of that double bond, that ene. We're getting an ene here now with the double bond between the 2 carbons. And in this process now, you're also going to strain this bond with the phosphate. So this is now going to be an energy producing species again. And then finally, the last step is this strained phosphate is going to come off and produce an ATP, and you're going to end up with pyruvate. And then pyruvate can be converted into that acetyl CoA and go into the Krebs cycle. So through this whole process, we've taken a glucose, well, we've taken a bunch of carbs, we've broken them down into their monosaccharides, and then glycolysis is taking glucose and breaking it all the way down and forming pyruvate. So again, I'm not going to ask you guys to draw any of these structures. I'm not going to make you memorize what enzymes do what and that kind of a thing. Again, it's understanding this overall process. You know, you might get a question like, what's the ultimate product of glycolysis? Well, that's pyruvate. You know, and those kinds of questions. But we said when we do that digestion, we not only end up with glucose, but we end up with some other stuff, right? So we end up with galactose. This is a galactose molecule. This is a monosaccharide. Galactose will get phosphorylated into glucose 1-phosphate, and then it will get converted to glucose 6-phosphate. And now it can just get inserted in that very first step, because that's where we started. If you go back to step 1, we produce this from the glucose. So you kind of have a side reaction here that's taking galactose and producing this as well. And then now that galactose can feed into the rest of these steps and form pyruvate. Does that make sense what I'm saying? So this goes in to this the glycolysis chain. It's feeding into that whole chain as well. And then fructose gets, here's a fructose. It gets phosphorylated into fructose 1-phosphate, and then it will be split into this dihydroxyacetone phosphate and glyceraldehyde. And then these can be converted, glyceraldehyde can be converted to this glyceraldehyde 3-phosphate. That's part of that glycolysis chain. And this dihydroxyacetone phosphate can also be converted into this. And so fructose also gets converted and feeds into that glycolysis chain. Any questions there? So our glucose is getting consumed down, and then our fructose and galactose get converted and kind of inserted into the process as well. Oops. Okay. Glycolysis is regulated by some inhibitors. In the very first step, hexokinase, which is our enzyme used in that first step, gets inhibited by glucose 6-phosphate, which is the reaction product. So if we go back to step 1, all the way back up here, oops, we have this hexokinase being the catalyst for this, and it produces glucose 6-phosphate. But as the concentration of this goes up, this actually acts as an inhibitor on this enzyme so that you don't have this runaway reaction here, just burning up all of your glucose into this format if your body doesn't need it. As this drops, then your hexokinase is going to be active again, and you'll start breaking down glucose again. Make sense? Step 3, the phosphofructokinase is inhibited by excess ATP and citrate. So again, if we go back up here to step 3, here is this phosphofructokinase here. As if your body has your ATP goes up, it will inhibit this enzyme from reacting. And so it will act as a throttle on this process as well. And then finally, step 10, the pyruvate kinase is inhibited by excess ATP. So if we go back to step 10 here, the pyruvate will inhibit this kinase that's forming the pyruvate. I'm sorry, excess ATP. So if you've got lots of energy in your body, then it's saying, let's throttle this down. We don't need more energy. That's essentially what that's saying. So high levels of ATP means I got a lot of energy available. I need to throttle down the whole overall process. Okay, so pyruvate is the ultimate product of that whole mess. And what does it get used for? Well, it can be converted, like we said, to acetyl CoA, which then goes into the Krebs cycle. So you have pyruvate basically forming a bond with this CoASH. Remember, it attaches to this sulfur. And there's a NAD plus that gets reduced in that process. And there is a enzyme that does this. And you're also going to get CO2 off as a byproduct. And so now this acetyl CoA can go into the Krebs cycle and be used to produce energy. Pyruvate can also be converted to lactate. And we'll see this in a bit, why it would want to do this. It basically will, if you have excess pyruvate, you can produce lactate, and then the body uses it elsewhere. And this, by the way, is a source of sore muscles. So if you go to the gym and work out and you're not used to that, you know how you get that horrible, my whole body aches kind of a thing. It's the source of that is lactate building up in your muscles, and that is come by the fact that you've converted this pyruvate in your body into lactate. So that's where you get soreness of muscles. Pyruvate can also be converted to ethanol. And I was wondering last time I taught this, does that make you drunk? I don't know, but but it can also be, so excess pyruvate can be formed off into ethanol in your body. Okay. So if you guys remember, we have our cell here, and inside of the cell, we have our nucleus. We have these mitochondria, which are our energy producing guys inside the cell. And then this light green area is that cytosol, which is just sort of the filler of the whole thing. So you can see here that we need NADH here. This product of NADH cannot be directly brought into the outer membrane of the mitochondrial, into that inner mitochondrial space. And so what has to happen here is a process that utilizes a glycerol phosphate shuttle. to get the electrons from the NADH into the system. So what we have here is we have in the cytosol a dihydroxyacetone phosphate, and this will oxidize a NADH to NAD plus, and you will lose that double bond oxygen, and you will get the two hydrogens put onto here. So you get the hydrogen off of the NADH, and another free proton gets combined right here into this molecule. This is a glycerol 3-phosphate. This can diffuse into the mitochondria in here, and then it will get converted back to this dihydroxyacetone phosphate again. But in the process, it's going to reduce the FAD to FADH2. And then this can diffuse back out into the cytoplasm out here, and this is a cycle that can happen over and over again. So this is the mechanism for getting the electrons that are carried by that glycerol 3-phosphate into the into this inner membrane space of the mitochondria. That FADH2 then, if you remember, that was part of our electron transport chain. We have the NADH gets oxidized to NAD plus and gives us free electrons in this ETC, and FADH2 gets oxidized to FAD plus free electrons, and those electrons go into that electron transport chain in order to do the work that we need done. I just grabbed this piece of a slide off of last week's slide to show you. So we're getting those electrons out through this process, and then we're going to get 26 ADPs from the electron transport chain. That's through the Krebs cycle. We will get 2 ATPs from ethanol. So there's where the ethanol ends up, is giving us more energy. We will get 2 ATPs from lactose for a total of 30 total ATPs from a glucose molecule. So we're getting 26 from the Krebs cycle, and then we will get 2 from ethanol that's formed by that pyruvate, or we get 2 from that pyruvate being converted into lactose. Any questions there? Okay, glycogen. You guys good with keep going, or you need a quick break, or just keep plowing? I'm good to go if you guys are. Okay, so if you recall, glycogen again is the way that our bodies store this excess glucose. It's just this branched polymer of all these glucoses all chained together. And it's how we store the product of our carb digestion in our body. It gets stored in the muscle and in the liver. So glycogenesis is the process by which we make glycogen from glucose. So we're going to add another glucose to this chain in order to store it in our bodies. And this involves 3 steps, thankfully not 10. So we have glucose 6P from that first step in the glycolysis. This was our very first product. If the body doesn't need this to go into energy production immediately, then it will be converted from 6-phosphate to glucose 1-phosphate with a mutase. And what we see here is that it flips these again. So it's this mutase. It's going to bring the hydroxyl group off of here, or it's going to bring the hydrogen off of here and put it on this oxygen up here. And it's going to take this phosphate ion and put it down here on this oxygen. So this goes from a 6-phosphate over to a 1-phosphate. Then the second step is the glucose 1-phosphate reacts with UTP, this is uridine triphosphate, to form UDP glucose. So here's our glucose 1-phosphate, and here they've drawn it out. Remember that P with a circle around it is actually this whole critter. And this is going to react with uridine triphosphate. So this is uridine with 3 phosphates on it. And it's going to peel off 2 of these phosphates. We see that here. And then this is going to plug into and connect. So it's kind of in effect almost forming a diphosphate bridge between this ribose sugar and your glucose. And then the third step is that UDP glucose, the bond gets broken and the glucose gets attached into the glycogen chain. And so what you see down here is this UDP gets cleaved off. I now have that as a product. And this UDP can be converted back to UTP to be used again. And then this glucose part of this just gets attached into the chain. And so we will go from glucose in to glucose N plus 1. So this glycogen is going to just grow by 1 glucose unit longer. This does require 2 ATPs to do. One initially to form the glucose 6-phosphate, and then another one to convert the UDP back to UTP. So this is an energy consuming reaction to form glycogen. So again, this is storing excess glucose into your body as glycogen. Glycogenolysis is the reverse of that. We're going to get a free glucose off of this chain. So we're going to say, oh, we need energy. I need a glucose. We're going to go get one off of there. And again, it's in 3 steps. It's essentially reversing the process. So we're going to take our glycogen with a free phosphate ion, and this is going to peel off one of the glucoses from the glycogen. So we're going to go from N to N minus 1, and we're going to have a glucose 1-phosphate. So that freed glucose will react with this phosphate to give us the glucose 1-phosphate. And then this gets isomerized into glucose 6-phosphate, so with a mutase. So this is just reversing that back the other way. And then we now have a glucose 6-phosphate, which will react with water to form glucose and a free inorganic phosphate. And it uses a enzyme to do this. Glucose 6-phosphatase catalyzes this reaction. So we end up with this 6-phosphate, and then that gets brought back to glucose again. So what we have here is we have glycogenesis is where um, we're building up the glycogen. Trying to see. Okay, so the glucose comes in as glucose 6-phosphate, gets converted to glucose 1-phosphate, gets converted into that UDP glucose, and then it adds that glucose to that glycogen chain. So you now have a glycogen with an N+1 here. But then the reverse of that is taking your glycogen and coming over here and peeling off one of those as glucose 1-phosphate, which will get converted to glucose 6-phosphate, which can then get converted back to glucose again. And then, of course, this glucose 6-phosphate is just the beginning of the whole glycolysis thing, so it could move on into there to produce energy. Any questions there at all? Okay, so then we talked about gluconeogenesis, so we're going to look at that next, and we're going to look at the Cori cycle. So gluconeogenesis, remember, it's creating new glucoses from other products. So this is a process where glucose is synthesized from non-carbs in our body. This happens when your diet is lacking glucose. Your glycogen stores get consumed in about 12 to 8 hours from exercise or fasting. So if you're out of glycogen, it can't peel off and get glucose anymore, then your body will try to make glucose through this gluconeogenesis pathway. And this, incidentally, is why intermittent fasting is so popular, is that people that do intermittent fasting will do what's commonly called a 16-8, and some people will even do a 24. This is 16 hours of fasting, 8 hours of eating, or 20 hours of fasting, 4 hours of eating. But you're basically hitting this 12 to 18 hour wall where your glycogen stores are going to be gone. And so you will start shedding weight as you are not, your body has to consume all of that glycogen for your energy in your body. If you're not eating, then it's going to dip in and burn up that glycogen. Glyconeogenesis, because you're building, costs energy. It's going to cost 4 ATPs and 2 GTPs. And the process is it's going to take a pyruvate. It's going to react it with ATP, GTP, and a NADH and water. And out of that, you're going to get a glucose. You're going to get 4 ADPs, because these 4, we're going to peel off 4 phosphate ions. So you're going to end up with 4 diphosphate instead of triphosphate. You're going to end up with 2 diphosphate instead of triphosphate here as well. So you're going to end up with 6 phosphate ions as part of this whole reaction. And then you will also have 2 oxidized NADHs to NAD+. This is the net reaction of all of this. Okay, so glycolysis is going down this way. So we have glucose being broken down into pyruvate. So it goes to 6-phosphate, then fructose 6-phosphate. Then we get this bisphosphate, then this phosphoenol pyruvate, and then ultimately pyruvate. Gluconeogenesis just reverses that whole process. And so it will take the pyruvate, it flips it into oxaloacetate, which then can be transformed into the phosphoenolpyruvate, which can then be changed and so forth and so on back up to a glucose again. So glycolysis, we take glucose down to pyruvate. Glyconeogenesis, we're going to take pyruvate and push it back up and form glucose. So the Cori cycle is just showing the relationship between these two things. Glycolysis, the breaking down of glucose, happens in your muscles as you need to do work. It's going to burn up these glucoses to get the energy out of it. You're going to end up with pyruvate that then goes into the Krebs cycle to produce that energy. If there's excess pyruvate, though, you're going to form lactate, and that's that source of sore muscles that we talked about. And this lactate will get shunted out of your muscles into your liver. And then this lactate in your liver will get converted back into pyruvate when your body needs to do this gluconeogenesis. So gluconeogenesis happens in the liver. You go through that process, you end up with a glucose, which gets shunted back into your muscles for energy again. So again, a good test question might be like, where does glycolysis happen? Where does gluconeogenesis happen inside the body? Or maybe what gets shunted from your muscles to your liver? Those kinds of things. Any questions there? Okay, and then finally we have this pentophosphate pathway that we mentioned at the beginning. So this happens in two stages. There's an oxidative stage and there's a non-oxidative stage. And in this non-oxidative stage, it's going to form a ribose 5-phosphate, which will get used in the synthesis of ATP, GTP, UTP. It can also form CoA. It can form NAD plus, and it can also form FAD. And this ribose can also be used to build RNA as well as other sugar phosphates. And ultimately, you can end up with this glyceraldehyde 3-phosphate that is a product of this whole thing. You end up with these two compounds ultimately, is a glyceride 3-phosphate and a 2 moles of fructose 6-phosphate. So the overall reaction here, the net reaction of these two stages, is we have a glucose 6-phosphate with a NADP plus and water, which gives you these 2 phosphates. You get some carbon dioxide coming off as well. You have a NADPH, and you've got 6 protons that come off of this reaction as well. I think I have a diagram of this. So yeah, so we're back here. This pentose phosphate pathway takes excess glucose and makes pentoses, which can then be used as nucleotides, or they can be converted back into glucose if the body needs it. So again, I think we're close to the end here. But just to go over this again real quick, we take in carbs, we digest them, we end up with glucose. Glucose goes through glycolysis to give you pyruvate, which then can get converted to acetyl CoA and feed this whole energy producing citric acid cycle. We get all these ATPs out of there. If you have too much glucose, then it can store it in your body as glycogen, these big branched polymers of glucoses, and this is called glycogenesis. If our body is low on glucose, we can pull it off of there through a process called glycogenolysis, and that will give us free glucose to then give our bodies energy if we need it. If our glucose is low, we can also use excess pyruvates to form glucose. And if we have too much glucose in our body, we can also create these pentose sugars, which can then be used to form nucleotides. Or if it drops back down, we can pull those pentoses back and convert them back to glucose. Any questions there at all? Pretty cool, huh, though? I was telling the last class that things like, before I started teaching this class, it's like I knew we had liver. I knew I had a liver. I didn't totally know what a liver does. I know that I have mitochondria in my body, but I wasn't totally sure what it all did. And I knew carbohydrate intake. Too many carbs is bad for you. Well, now I know why. So, it kind of explains a lot of those types of things. Okay. This pentose phosphate pathway, I think I already covered this. If we have a high demand for ATP, then this pathway, go back to this pathway here. If we need energy, then it will go feed back into this glucose and this glycolysis pathway to give us that energy that we need. If we have a high demand for this NADPH, this is NAD with a phosphate and a hydrogen on it, then it will recycle the intermediates of that to glucose 6-phosphate and thus give us more NADPH. So it will form more of this, which then can react and give us more NADPH if it's needed. This is this pentose phosphate pathway we're talking about. And then if we need components to build nucleic acids, then this ribose 5-phosphate will go, the pathway will stop, and it will leave us with the ribose 5-phosphates in order to form those nucleotides. So if our body needs these nucleotides, then it's going to use this pathway to form these pentose sugars, which will go into the nucleotides that way. There are hormones that regulate carbohydrate metabolism. Insulin, which we've looked at before, is a 51 amino acid protein that gets produced by the pancreas, and it will take up excess glucose in our bodies. Glucagon is another hormone. It has 29 amino acids in it. It's also produced by the pancreas. and it gets released when glucose blood levels are low. It therefore speeds up the glycogen conversion to glucose. So it's kind of the opposite of insulin. Insulin's going to help us store excess glucose, and glucagon is going to cause us to peel off and burn up the glycogen because our bodies need energy. And then finally, the hormone epinephrine, which is also called adrenaline, which gets released in anger, fear, or excitement, will stimulate the glycogenolysis in the muscles and give us quick energy. So it's going to cause your muscles to burn up those glucoses. Get back there. Where is it at? It's the consumption of the where we get glucose from the glycogen. And so epinephrine will accelerate that process so that we're getting those free sugars to then pump into our bodies to get energy. And this is why people have sudden burst of energy with in places of anger, fear, and excitement. And we are at the end. I didn't get a chance to put problems in here, but I will do that tonight since we're getting done a little early. But before we go on to that, are there any questions about anything we covered tonight? Looking at our syllabus. So we just did today chapter 13. Next week we will do chapters 14 and 15. They're both shorter chapters. And we will do a review, getting ready for exam 6. And then the following week will be exam 6 that you all can take while I'm in Georgia. Yeah, I'm looking forward to some time off. Okay, well, if nothing else, yeah, so the last exam will be 12 through 15, right? We just did 9, 10, and 11, so 12 through 15 will be the last exam. And next week we'll do a review of, I guess we're doing a review of the whole test. Okay. Oh, I was going to put it at the end of the class. That's right. Okay, I remember now. Okay. If nothing else, we are